this oral presentation is micro-assisted solvent extraction of three important Garcinia. These are the general outline of my presentation. As I've been working on Garcinia species, mainly Garcinia pedunculata, Garcinia indica, Garcinia talbuti. There are worldwide 180 species which South America and in India also. Is well known for hydroxycitric acid, which is a proven natural anti-obesity agent, and there are many biologically active compounds isolated from the various parts of this tree, and also contains it also possesses so many pharmacological activities. Hmm. These are three Garcinia species. First one is Garcinia pedunculata, second one is Garcinia indica, and third one is Garcinia talbuti. Uh, Garcinia pedunculata is found in Assam, which is also commonly known as Brochegra, and second one is Garcinia indica, which we are using in kitchen also to enhance the taste of the uh, and third one is Garcinia talbuti, which is known as limboti, and in Marathi it is known as limboti. Now, to extract biologically active molecule, we use generally Soxhlate or heat reflux method, but the conventional method having so many drawbacks like it takes longer extraction time, more organic solvent consumption, lower extraction efficiency. It is the process is also tedious, and it may destroy your biologically active molecule. Now, to overcome this problem, we use modern extraction technique, which is microwave assisted solvent extraction, which is which saves our time. It consumes lesser organic solvent. The efficiency of the extraction is also high, and the cost is less. Now, I am focusing on phenolic compounds. Phenolic compounds are uh, compounds which possess more than one aromatic ring and more than one hydroxyl group, which are, and these phenolic compounds are widely found in plant kingdom. It also possesses so many biological activity. To extract such phenolic compounds, we use organic solvents like methanol, ethanol, acetone, ethyl acetate, hydroalcoholic, water, or acidified organic solvent. Now, this Garcinia pedunculata, Garcinia indica, and Garcinia talbuti were collected from the Assam Agriculture University, Jorhat, and Garcinia indica and talbuti were collected from Konkan region of the Maharashtra in July. Study was done in our lab, Applied Chemistry Department, SVNIT Surat. We use microwave reactor rotosyn milestone microwave laboratory system. This is general procedure of micro assisted solvent extraction. We have fixed 3 times 5, 10 and 15 minutes, and power is 100 watt. We are taking 1:10 ratio, 5 gram of powdered plant material and 50 ml of solvent. We are using 6 different solvents with some are polar, some are non-polar, and other are moderately polar. So we are repeating this in 3 cycle, and after extraction we are getting filtrate which is dried under vacuum. This is the conventional method in which we are refluxing the plant material with appropriate solvents for 6 hours and dry it and getting percentage yield. After extraction, we have done total phenolic content estimation using Folin Fiocalte method described by Singleton and Rossi. And in this method, the extracts were prepared in methanol. The concentration was taken 100 ppm. Then mixing with polyethylene reagent and distilled water. After adding some more reagent, we recorded we record its absorbance at 75 nanometer. This is the yield we are getting with the help of microwave. In methanol and water, we are getting more yield compared to other because of microwave absorbs the polar organic solvents. Polar solvents absorb microwave and non-polar which are very poorly absorbed, which absorb poorly. This is for Garcinia indica fruit print, in which we are getting 69% at only 5 minutes. Okay. And after 15 minutes we are getting 78% of yield. This is Garcinia talbuti seed powder we have taken, in which we are getting more yield in water, polar as well as methanol water. We are getting also around 50% of yield. In ethyl acetate and other non-polar solvents, we are getting less yield. This is the yield comparison. This block is this column is of heat reflux method. After 6 hours, we are getting only 9%, 8%, around 50%. But in just 15 minutes, we are getting more yield. This is total phenolic content in all the 3 plants we have compared with this with heat reflux extraction conventional method. We are getting more in hydroalcoholic solvent. We are getting 50 around 50 milligram per gram total phenolic content, and in non-polar the phenolics are not not extracted in non-polar solvent. We are getting less phenolic content. This is this big blocks of total phenolic content of Garcinia pedunculata. Conclusion: the extract yield was determined using greener approach called a micro-assisted solvent extraction. In all Garcinia species, methanol, water and hydroalcoholic solvents, we are getting more yield. Non-polars which poorly absorb the microwaves, we are getting less yield and less total phenolic content. So MASC micro which is helpful to get better efficiency of extract, and as well as phenolic content we are getting more in that. And 3 solvents, methanol, water and hydroalcoholic solvents are preferable. These are major references. I am thankful to my esteemed and learned guide Dr. Premlata Kumari and Dr. Satyan Sukumar. I am very thankful to SVNIT Applied Chemistry Department, Organizing Committee of Indian Science Congress, and also SV University Tirupati, Andhra Pradesh.